Welcome, everybody. This is Aga, your host of Life Science Track. Let's delve together into the life science adventure. We are going to start with a zebrafish, an important and widely used vertebrate model organism in scientific research. Did you know that the fish got its common name from the presence of 5 uniform pigmented horizontal stripes on the side of its body? Yes, it is. Now we are going to hear more and about the shiny app that Unravel Zebrafish hot intercellular communication. Please join me in welcoming our first speaker, Pratik Arora, Bioinformatics Research Associate from University of Bern. The floor is yours.
Hello, thank you, Aga, for the beautiful introduction to me as well as the Zebrafish. Well, so I'll start, however, on a bit darker side. Um, as we confront the WHO data, which it releases every year, you can see that one of the dark pictures that it raises out is that the leading cause of death globally is actually an ischemic heart disease. Um, it's surprising to note that it keeps on increasing every year, and you can see that it already from 2000 to 2019, that number of deaths have increased up to 9 million. Thus, it's obvious that something needs to be done about it. One of the major causes behind these deaths after an ischemic heart disease is the myocardial infarction. And unfortunately, when an infarction happens in a human heart or a heart attack, as we might say, um, it it causes a fibrotic scar, and this scar never heals in a human. However, in the animal kingdom, this is not really true for all the species. For example, mice, when they are just born, or neonatal mice, have the ability to repair the scar and regenerate their heart. There's one more species, one more animal, which is much more interesting, which is a zebrafish, which can repair its heart all throughout its lifetime. And that's why we are focusing on this animal today. So, what is this animal? So, this is a picture of how it looks like, and it's a very interesting model organism found in the Southeast Asia, for example, India, Bangladesh, etc. And it's important to note that this has got all the main organs, has a 70% all the 70% of the human genes have zebrafish orthologs. It has a fully genome which is sequenced, and it's easily genetically manipulated. A lot of disease models have been made on that, thus making this organism a right choice to do basic researches based on which we can further transform into multiple translational research. So, for as I said, not just as I said that already, that it can regenerate its heart, but not just the heart. It can also regenerate other organs such as liver, pancreas, skin, fins, spinal cord. And this organism is not just some random organism, but it's actually one of the major model organisms to be used. For example, more than 60,000 articles have already been published on zebrafish research. But coming back to the topic of our interest today, which is the heart, in which we perform the regeneration study. So, to perform this regeneration study, we use cryo-injury model, in which means that we freeze a part of the heart, ventricle of the heart of the zebrafish, and within a few days it regenerates. So, as this injury is performed, you can already see that it causes a lot of damage to the heart. So, for example, there's first the inflammation stage, or basically the first basic. Hello, hello. Can you hear me? Yeah. Okay. Sorry. So it seems it's okay. Yeah. Okay. So so it already seems that the first thing that comes out is the inflammation, which is basically the injury response, in which multiple immune cells come on, and then you have these fibrosis, or a scar is formed around the heart, around the injury area to stop its more progress, and then the extracellular matrix, or it starts to heal, and by already 30 days or 1 month, the cardiomyocytes or the heart cells start to proliferate and start to regenerate the whole heart. Now, as you can already see, probably from this schematic, which might be too complex, but that there are multiple cells which are involved in this whole process. And we in the lab had already identified a few of the cell populations which are important for the regeneration process. And once this, and we now wanted to understand that of these critical populations that were identified already, how do they communicate with each other? This work that I'm showing now was also done in collaboration with the PhD student in the lab, João Carvalho. And to answer this, we made a Shiny app. I'll just go through briefly through this app, where basically we made a network of various ligands and receptors, which you can see over here. When we zoom in, you can see various molecules and ligands and receptors which are formed over here. Each of these nodes or each of these points represents a molecule which you can highlight and see what it's interacting with, what our receptors are. And then if you go down, you have a table which has all the details, such as the gene symbols, the cell type from which it's interacting, the score, various genes that it influences, or the direction, whether it's a source or ligand. You can look at how much it is known in the literature, what its rank is in the network, and what its source in total is. You can look at these parameters also as a graphical parameter, highlighting individual parts. And then you can also look at the other visualizations, such as this one, in which you can see various other details, a table, a much simplified table, and overall the whole app. So what does this table act? What do these tables and app really represent? So coming back, so for this, we have to come back to the figure that we already saw, that we are trying to understand the intercellular communication. The data behind this app is actually a bulk RNA-seq from fact sorted population. Okay, what is these too many words now over here? So what is bulk RNA-seq? So you have the heart, which is there. So what we are doing over here is we are taking out cells of our critical point, which are critically important to the to the population, to the regeneration, and you can get them out. You can sort them. With each cell, you can take out the RNA, which is the main messenger for all the proteins which are made in a cell. Then you take all the RNA, which is the bulk RNA-seq from all the cells together, and you have a bulk RNA-seq from fat sorted area. This complex data, when taken from multiple studies, can pose certain problems, such as batches. So what do I mean by that? So certain cells, such as the fibroblast or the other cells, now, as you can see over here, are presented in various different populations that they separate out. But we overcome this by using batch correction techniques such as combat-seq, and now we make these data comparable to each other, where all the fibroblasts come together, cardiomyocytes come together, and endocardial cells come together. Using this, we then went on to select the ligands and receptors, which we assume is the main source for mediation of the intercellular communications, which you can assume as some keys and locks, which will work together to form a signal, or to activate or switch on a signal. For this, we use the Omnipath database, which has experimentally validated ligands and receptors, and we converted them from humans to fish in the Omnipath. Using this, we also performed a differential analysis in which we tried to see whether the genes which are activated or deactivated in 7 days, which is the regeneration time, versus a condition where there's no regeneration, which is like 0 days. Thus, to get like, okay, these genes are the ones which are activated, and these are the ones which really come across while and responsible for the regeneration. Having done this, we create a ligand receptor network in which each of the color represents a cell type, the 4 cell types that we talk about, and up and down is represented by the size, and squares and circles represent whether a gene is upregulated or downregulated. Looking more into the detail, so for example, this is a node in the network. You can see this, which the network which was made using iGraph. So each of the node represents a cell type plus a ligand or a receptor. The color is as I said already, is represented by the cell type. The label represents whether it's a ligand or receptor. The size is represented by the log to fold change. The edge of each of these nodes are represented by the ligand receptor communication, which we got from the Omnipart database. The following was imported into the Viz network library, and we used the layout from the iGraph to keep it constant throughout with a set seed already, and we allowed the highlighting of the various nodes to allow the and answering and visualization of the neighbors that are together. So neighbor here would be if it's a ligand node, then all the receptors will be highlighted, and if it's a receptor node, all the ligands will be highlighted. To allow for more conversion, we also went on for multiple parameters scoring, such as differential expression, page rank, literature search, and differential genes and interacting partners. Altogether, we saw that this gives us a ranked list of the network, and a lot of them were actually already identified, giving us a hope that we can identify actually even more from the unknown list that we have got. One of the such examples is NTN1A. And coming back to the Shiny app, we also provide a table to look at all of this data that I talked to you about in a format which is rendered by the data table library in the Shiny app, which represents score, gene symbols, with gene symbols from human, which we converted, log to fold change, influence genes, the direction, the literature, the page rank, everything is combined over here. The same data is also, if you do not want to read all through this can be difficult to understand what the score is. So for example, for NDN1A, you can look at it in the graphical format below, which is now arranged in alphabetical manner, and you can see that, oh, okay, this green one is let's say, or heart one is heart literature or something else, or page rank, what is contributing to the whole score and giving the rank of the whole network. You can subset this data. So for example, if we subset NTN1A, this allows for subsetting of the allows focusing on the network, subsetting of the data in the data table, and subsetting of the graph itself. So thus giving a full comprehensive analysis of the whole table. Okay, some might find that, okay, this visualization is way too complex. There are too many nodes over here. So the graph, we also offer other visualization wherein just the ones which are really highly differentially expressed, we plot them as a scatter plot with X and Y axis as literature searches. The data table here only represents highly differential genes, giving options for subsetting. And the color is represented by log to fold change. The shapes again represent the cell types. The page rank is now given by the size. It also allows for tooltips where you get all the data on the app itself, on the graph itself, and you can also subset this for interspecies comparison. Here we have done it for one of the mouse species, which is from Wang et al. You can also download the data if required. So to summarize, we found a novel way to find preclinical targets in the heart regeneration, and we are now actually we also present this data with a user-friendly interactive app to help the exploration of the data. The candidates from all these ranked lists are now being experimentally validated. We are using full body and tissue specific knockouts using CRISPR-Cas9, and we are studying the effect on the regeneration, and we have all of this with the manuscript in preparation. And this, I want to thank all the team that was involved in this, especially Professor Nadia Myocardial, whose group I am in and who gave me the opportunity, and the PhD student who I worked on this project, Joao Carvalho. I would also like to thank the various funding agencies, that is the European Union and the REANIMA grant, and you for your attention. And I'd be happy to take more questions.
Thank you so much for the presentation. We do have some questions to you. First question, is there a GitHub link to access the source code of the app?
Uh, Not right now, but so because we are still in the paper is in revision right now, but hopefully soon we'll have the GitHub online. I will post it on LinkedIn and Twitter because this data is right now not published, so not right now publicly available. But I can share with you. You can DM me or like email me. We can talk about it. Yeah.
Mhm. Thank you so much. Is PageRank the eigenvalues of the adjacency matrix or some other algorithm?
So PageRank is actually the algorithm that actually made Google Google. It takes into account how many neighbors are talking to one node, and as a result of that, what node is the important one. It's something different than um, the other network parameters such as the betweenness or the centrality, but it's not really eigenvalue matrix, or it's another algorithm basically. Yeah.
Okay, thank you
Mhm.
so much. One other question. What do you think is the most important in visualizing complex data in Shiny app? Maybe you can share some tips to our audience.
Yeah. So for me, for example, the most complex thing here was the interactivity between the network, um, the data table, and the graph. All of them to subset all together, and just making sure that this is one of the things that only the thing that you subset for or want to focus on was quite tricky. Um, so to do this, I had to move out of Wiz network. We had to um, I had to develop a technique to basically put this in separate vector, then use the same vector and put it in Wiz network, and then put it across, subset it using deployer and on all using on all three different datas, rather than, even though Wiz network does allow focusing, but just using one of them to focus all three was quite tricky, and that was something that was yeah, important, I guess. Yeah. Yeah.
Okay,
Yeah.
and did you need to learn JS to do that too?
No, actually, no. I actually only know. I only use the Wiz network and the Shiny apps and R basic R. That's all. Yeah. So no, no, I did not have to use JS or anything. Yeah.
Okay. Maybe you have some resources or tips where to find how to, when you are learning about iGraph and other network visualization. If someone is interested, where find the first starting point to look for?
So I think the official documentation by iGraph is quite well documented. Um, it was quite helpful, and I just used that one for iGraph. However, what was it did take me some time to understand really was that iGraph can really take data frames itself as the full uh, annotations, so-called, or basically the one-to-one allocations for all the nodes. So that was really one thing that I used quite a lot over here. um, the functionality of iGraph from to convert data frame to network itself was also pretty good, because the Omnipart database was basically just a table, and to convert that from ligands to iGraph was pretty good. But all of this was quite well documented in iGraph, and I'm really happy that was really there. Similarly, Wiz network was also well documented, and it was quite straightforward actually to do this with the official documents, I must say. Yeah.
Okay. A final question. What was the most challenging part in developing this Shiny app?
Um, so as I said, that most challenging part was getting the data for this, basically, to analyze the data which is behind it, because the one of the one of the challenges was that the database for the ligands and receptors was human based. However, we have Zebrafish as a model. So to convert this Zebrafish to human was not so straightforward. We spent more than 2 months just trying to convert it. Finally, we decided to convert the whole database to Zebrafish rather than Zebrafish database. But that, but once we were through with that, it went quite ahead, good ahead, and the app itself was then quite straightforward with all the data in hand. Yeah,
Okay. Thank you so much for your
yeah.
presentation and all the
Thank
answers
you.
for the question. Congratulations and thank you so, so much. Have a good
Thank you for the opportunity.